Hello, this is Mr. Watkins. You are getting ready to watch another video lecture. This particular video lecture deals with how materials move across a membrane using particular proteins. This diagram, or something very similar, can be downloaded off of MrWatkins.com. First, let's go over some basic term and kind of refresh your memory. Anytime you have movement from a high concentration of particles to a low concentration of particles, that's called diffusion. So here in this first example on this left-hand side, we have a large number of particles on the outside of the cell, a few on the inside. These are very small particles. The key indicates they're oxygen, and they will diffuse straight through the membrane. This is called simple diffusion. Simple diffusion. Now, the next part of this membrane has a membrane protein. This membrane protein, because it's touching the inner layer here, would be an integral protein. Any protein that touches the inner layer is called integral. And because the protein goes across the entire membrane, it's also called a transmembrane protein. Again, we have particles on this side, high concentration, particles on this side of the membrane in a low concentration. These particles move through this inside basic hole or tube and to get to the other side with diffusion. Now, because of this hole or this tube, we call this a channel protein. It also could be called a porin or a pore. And bottom line here is that we still have diffusion. We still have from high concentration, movement of particles from a high concentration to a low concentration. They're just being helped across the membrane because of this channel protein. This would be facilitated diffusion, because it is being helped or facilitated across the membrane. Now on our key over here it says these are polar molecules. Polar molecule, if it tried to go through the membrane, would get stuck right here in this inner part. Remember that your membrane is made up of very small phospholipids, a phosphate head that's polar, and a tailpiece that is made out of fatty acid, part of the lipid part, that is nonpolar. So these polar molecules would get stuck, would not be able to make it all the way through. They would be repelled. And so because of this channel, these molecules can go through as long as this channel on the inside is polar. Now, the next diagram is basically the same thing. We're looking at facilitated diffusion. But again, if you look at our key over here, these molecules are water molecules. So we have a special term for when water molecules move from a high concentration to a low concentration through a membrane. We call that osmosis. And again, we have a tube or a pore running through this protein as this protein spans the membrane. This particular protein, the one that's involved in the movement of water, is called an aquaporin. And that's just a very special name for this particular channel protein that's transmembrane, and it's also integral because it touches the inside. Now, before we get any farther, these integral proteins, if they have a piece, and obviously to be integral they have to have a piece, but the piece that touches this inside part of the membrane here on the outside has to be nonpolar. Remember that nonpolar likes nonpolar and polar likes polar. So these sections out here and also on the inside would need to be polar because they're dealing with water or with the polar molecules that are either outside or inside the membrane. Our last diagram here is a diagram of a carrier protein or carrier molecule that carries the materials that the cell wants across the membrane. And I want you to notice that this has a low concentration on this side and a high concentration on this side. And the direction that we're showing is that it's going against the concentration gradient. This would not be diffusion. This is something the cell is going to have to spend energy in the form of ATP. The cell will have to spend energy to go against the diffusion, against that concentration gradient. Carrier protein. It's still, in this particular case, a transmembrane protein. It is also an integral protein because it does touch on the inside. Now, to do this, the cell spends energy, and when the cell spends energy to get something like this to happen, to get this material on the outside that's in a low concentration inside the cell, going against the concentration gradient. Cell uses energy. That is called active transport, the type of transport. Now, this particular diagram, the aquaporin, where we have a channel protein going with the concentration gradient, no energy being used. This diagram, again, going with the concentration gradient, cell is not using energy. And even here, where we have just simple diffusion going through, these three things are called passive. The type of transport is passive transport. In other words, there isn't anything the cell has to do. It's passive in its ability to get the membrane, get the materials across the membrane. If the cell wants to take in a large amount of material, like food, or a large amount of liquid that's outside the cell, we have another process that involves the membrane, and that's called endocytosis. And there will be a separate lecture for this particular piece. Again, just to reiterate, simple diffusion, passive transport, we're moving with the concentration gradient, no help from a protein. Facilitated diffusion, passive transport, we're being helped by a channel protein to get the materials across the membrane, otherwise they would not be able to go. Osmosis is the movement of water from a high concentration to a low concentration, and in this case we are using an aquaporin, the special channel protein that moves water. And then of course we have an active transport situation on the end here, where a carrier protein is going to carry the molecules against the concentration gradient once it's picked up enough energy that the cell has to give to get this to happen. Again, the diagram can be downloaded off of MrWatkins.com. Thank you.